Hello everyone, welcome to Teaching PATHSHALA. Today we are going to discuss about the prevention of polyspermy in sea urchin. In the previous video, we have studied about the fertilization of sea urchin. So if you have not seen that video, I will mention the link in the description box as well as in the end of this video. So the prevention of polyspermy in sea urchin has been taken from the unit of developmental biology for the preparation of CSI and NET. In the end of this video, we are going to solve some CSI and NET question on the prevention of polyspermy. Along with this, I will provide the link of the playlist that will contain all the different videos from developmental biology unit, which is important from CSI NET exam point of view. We are going to start with what is polyspermy. So the entry of a multiple sperm in a single egg is known as a polyspermy. Poly means multiple, spermy means the process through which a sperm is going to enter. That is why the whole process is regarded as a polyspermy. So in normal process, there is a fusion of one sperm with one egg. But when there is a case of a polyspermy, there is a fusion or the entry of a multiple sperm inside a egg. And although this condition is not at all normal, because this is going to lead to a disastrous consequences in the most organism. If there is a process of polyspermy that has been occurred successfully, so obviously instead of a zygote that is a diploid, there will be a formation of a triploid or the tetraploid type of a zygote. Okay, and in this case, during mitosis, some cell will going to receive an extra copy of certain chromosome and other cell will lack them. So this overall thing will going to generate a very abnormal offspring. That is why the prevention from the polyspermy is very necessary. This prevention of polyspermy in sea urchin, they mainly used to occur in total two steps, or you can say that total two way. Okay, the first way is the fast block of polyspermy. The fast block of polyspermy is achieved by changing the electrical potential of a egg plasma membrane. And the second process or the second type is the slow block of polyspermy. The egg of sea urchin and many other animals have a second mechanism to ensure that the multiple sperm do not enter the egg cytoplasm, which is also called the cortical granules reaction. After the successful fusion of sperm and egg, there is the occurrence of a fast block of polyspermy. So after the fusion, just after the 3 to 4 seconds, there is occurrence of a fast block of polyspermy, and this is the first type of blockage. And after the completion of a fast block of a polyspermy, second time there is a slow block of polyspermy is going to occur. So in a single process, this two type of a blocking system is going to occur. Okay, it's not like that one organism is going to represent a fast block of polyspermy and one is going to represent a slow block of polyspermy. No, this is going to occur at the same time. The first occurrence will be going to be of a fast block of polyspermy, and after the completion of a fast block of a polyspermy that is mainly used to take a 1 minute, there is a starting of a slow block of polyspermy, which is also known as a cortical granules reaction. Now there is a question that what is the need of this two type of blockage? Because if there is already a one type of blockage, that is the fast block of polyspermy, what is the need of the slow block? Because both are preventing the polyspermy, so one type will also be enough for the blockage of a polyspermy. So the reason behind this is that the fast block is mainly used to occur for a transient time. Whenever there is a fusion in egg and sperm, just after 3 to 4 seconds, the fast block used to activate themselves. This pathway will going to activate themselves, and there is a change in the overall electrical potential of a egg plasma membrane. But this change is reversible. Okay, this change is only going to take place for only, I think, 50 to 60 seconds, and just after that it will revert back to the normal condition. So the chances is that if there is still some sperms are there which used to bind to the egg surface, then after the reversal back of this electrical potential, they are going to make their entry inside a cell, and there will be again a occurrence of polyspermy. So for preventing that attack by the sperm that is already bounded to the egg surface, the second step of blockage will going to take place, that is the slow block of polyspermy. And that is what mentioned in the green highlighted portion. The fast block to polyspermy is transient, since the membrane potential of sea urchin egg remains positive for only about a minute. The brief potential shift is not sufficient to prevent a polyspermy, which can still occur if the sperm bound to the vitelline envelope are not somehow removed. This slide I have taken from the previous video just to show you that where this blockage is going to take place. So this circle which you can see in the red color, that is the area where this polyspermy blockage is going to take place. Now let's see the concept behind the fast block of polyspermy before moving to the steps that are there in the fast block of polyspermy. So the concept is that initially the egg is in a resting membrane potential. That means inside a egg the membrane potential value is minus 70 millivolt. And the students who don't know the meaning of potential, so it is the energy which is required to separate the two charges are called the potential. So in the condition when there is absence of a sperm, at that time the membrane of our egg, you can say that they are in a resting membrane potential by showing a value of minus 70 millivolt. And this negative charge of the egg membrane is due to the presence of a potassium ion inside a egg. And this is the reason for the negative charge of a egg. So you have to understand that before the entry of a sperm inside a cell, inside a egg cell, the egg is having a negative charge on its surface, and this negative charge is due to the presence of a potassium ion inside a egg cell, and that is responsible for its resting membrane potential of minus 70 millivolt. Now let's talk about the second condition when the sperm makes a entry inside a cell. Then what happen? The membrane potential changes to the plus 20 millivolt, which is called the depolarization. This change is due to the sodium ion influx inside the egg cell. Now egg membrane is positively charged. So initially without the sperm the egg is having a negative charge, but after the entry of a sperm now the egg will going to have a positive charge due to the positive membrane potential. And what is the reason behind this positive membrane potential? You know that in a sea urchin there is a presence of a external fertilization. That means the egg and the sperm both used to meet inside a water, okay, mainly inside a ocean, because they used to follow a external fertilization event. So during this process, there is a huge amount of a sodium that is present in a sea water. So whenever the sperm tries to make an entry inside a egg cell, at that time some amount of a sodium they will also try to enter inside a egg, okay. And after the entry of that sodium ion, the overall membrane potential of egg is going to shift from negative to the positive, because sodium ion is having a positive charge. So if the sodium ion is going to make an influx inside a egg cell, at that time it is going to lead to the change in the membrane potential to plus 20 millivolt. Now let's talk about the third concept. As because initially the egg was having a negative charge. So what is the reason of binding the sperm to this negatively charged egg? So the concept is that the sperm is having a positive charge on its surface, which is going to bind to the negatively charged egg. And the main reason behind the positive charge of the sperm is that it has some positively charged amino acid or protein that is going to give a positive charge nature for the sperm. And the negative charge of the egg is due to the negative membrane potential due to the presence of a high potassium ion. So we have seen the total three concepts. First concept is that the egg is having a negative charge and the membrane potential is mainly the minus 70 millivolt. Second concept was that after the entry of a first sperm, the membrane potential get changes from minus 70 to the plus 20 millivolt. That means now the egg is having a positive charge on its surface. Third concept was that initially the sperm is having a positive charge. That is why it is attracted to the negatively charged egg. Now let's move to the actual step of fast block of polyspermy, which is also known as the electrical changes. Now let's start with the first step. The egg cell membrane initially have a negative charge. This negative charge will attract the positively charged sperm. So now you know the reason behind both of them, that why the egg is having a negative charge and why the sperm is having a positive charge. The second step is that after the entry of a sperm, the ion concentration of an egg get changes. And you know the reason. Now let's move to the third step. During the entry of a sperm, the influx of sodium ion in the egg from the sea environment will take place by the opening of a sodium ion channel. The fourth step, as a result of the K plus ion, efflux take place. Okay, when there is an entry of a sodium ion inside our egg cell, along with that there is some amount of a potassium ions too, which used to efflux from the egg membrane. Now the cell membrane charge changes from the negative to the positive due to the influx of sodium ion and the efflux of a potassium ion. This you should remember that there is a high concentration of a sodium ion which used to influx inside a egg, okay, but the efflux or the removal of the potassium ion is somehow very low as compared to the influx of a sodium ion inside a egg. The next step is that now due to the positive charge of egg, no more sperm is going to bind to the egg surface, and finally the fast block of polyspermy is going to take place. So the actual concept is that initially due to the negative charge of egg and the positive charge of sperm, there is an attraction between both of them and the fusion will take place. But just after the entry of a first sperm inside a egg, there is a membrane potential changes, that is the shifting of a membrane potential from negative to the positive. Now due to this shifting, there is a positive charge on the egg surface, and now the other sperm is also having a positive charge. So obviously the positive charge and the positive charge is going to repel each other. So in this way they are going to prevent the fusion of some other sperm with that egg. And this is the whole concept behind the fast block of polyspermy, that after the entry of the first sperm, the overall membrane potential of the egg get changes from negative to the positive, and now the positive charge of sperm and the positive charge of egg is going to repel each other. In this way there is a prevention of the polyspermy in the fast way. And this is also called the electrical changes, because there is an involvement of a different ions which is going to take part in the changing of the membrane potential. So the conclusion from the previous slides is that the negative membrane potential, which is having a minus 70 millivolt value, sorry, this should be a minus 70 millivolt, not volt. So the negative membrane potential, which is represented as the minus 70 millivolt, this is going to favor the fertilization between the sperm and the egg. But when the positive membrane potential is there, that is the plus 20 millivolt, at that time there will be the no fertilization, that means the prevention from a polyspermy. So the negative membrane potential is going to favor the fertilization event, whereas the positive membrane potential is going to favor the prevention from the polyspermy. The third case is that if the supply of sodium ion is not sufficient to cause the positive shift in the membrane potential, then in that condition the polyspermy is going to occur. So if we want to make a key point from the first block of polyspermy, we can say that it is a transient block which used to occur just after the 2 to 3 seconds after the binding of sperm with the egg. Okay, and it used to last for at least I think 40 to 50 seconds, or maximum it would be a 1 minute. Next, it used to trigger a electrical response inside the egg membrane. It used to open an ion channel, and finally the sodium ion is going to change the membrane potential. Now let's talk about the slow block of polyspermy. So this slow block of polyspermy mainly used to take place I think 30 to 60 seconds after the first successful binding of a sperm with a egg membrane. So it is a calcium ion mediated blockage. So calcium ion is involved in this whole process. It is the second stage of prevention from the polyspermy, as well as it is a permanent, although the first block was only for the transient. The next step is that the cortical granule reaction, a slower mechanical blockage to polyspermy that become activated about 30 to 60 seconds after the first successful sperm egg attachment. And the last point is that it is the action of a cortical granules. So from the figure you can see the location of a cortical granules, that they are just placed I think after the vital line membrane. Now let's move to the slow block steps of polyspermy, which is also known as the cortical granules exocytosis. So directly beneath the sea arching egg plasma membrane, there are about 15,000 cortical granules. Okay, so this is the location of a cortical granules. Upon sperm entry, these cortical granules fuse with egg plasma membrane and release their content into the space between the plasma membrane and the fibrous mat of a vital line envelope protein. So it is regarded as a slow mechanical block, and this involves the several protein that is going to released by the exocytosis of a cortical granule, and this is going to make a change in the vital line membrane of a egg, and finally this is going to make the hardened fertilization envelope, which is going to repel the overall sperm that has been bound to the egg cell surface. So let's first talk about these steps. So after the entry of a sperm and egg, there is a several protein that are released by the cortical granules exocytosis, and the first protein is the protease. And the function of this protease is that it used to dissolve the protein that used to connect the cell membrane and the vital line membrane, as well as it used to clip off the binding receptor and any sperm that is attached to the egg surface. The next factor that is used to secreted from the cortical granules exocytosis is the mucopolysaccharides. And the function of this mucopolysaccharides is that it used to produce a osmotic gradient that is going to be responsible for causing the water to rush into the space between the plasma membrane and the vital line membrane. And this is going to cause the envelope to expand and become fertilization envelope. The third protein which is going to release from the cortical granules is the peroxidase enzyme, and the function of this peroxidase is that it used to harden the fertilization envelope by cross-linking the tyrosine residue on the adjacent protein, and in this way the bounded sperm is going to release from the envelope. The fertilization envelope start to form at the site of sperm entry and continue its expansion around the egg. As it forms, bound sperm are released from the envelope. This process starts about 20 seconds after the sperm attachment and is completed by the end of the first minute of fertilization. And the fourth protein which is going to release from the cortical granules is the hyaline. So it is have the ability to provide a support for the blastomere during the cleavage process. This is the diagram which is showing overall process that we have just described in the previous slide, that how the cortical granules is going to make the exocytosis of the different factor that is there in it, and after the release of this factor they are going to perform a different function, like there is a release of the hyaline enzyme, then peroxidase enzyme, then there was a release of the mucopolysaccharides. So these all factors is going to perform the different function. And in the second image you can see that there is a huge amount of sperm that has been bound to the outer surface of the egg. But after the release of these cortical granules factors, there is a change in the overall vitelline membrane and the plasma membrane, and this outer layer is going to develop into the hard fertilization envelope, and this hard fertilization envelope is now going to repel the overall sperm that was bound to the surface of an egg. So in this way, there is a occurrence of the slow block of polyspermy. So till now we have seen the procedure that how the slow block of polyspermy will going to take place. Now let's talk about the reason behind this slow blockage. Okay. So I think in the introductory portion of the slow block of polyspermy, we have mentioned one line that it is the calcium mediated slow block of polyspermy. Okay. So calcium is going to act as the initiator of the cortical granule reaction. In the previous slide, we have only mentioned that after the binding of a sperm and egg, there is an exocytosis of the cortical granule. And the reason behind that exocytosis of the cortical granule is the presence of a calcium behind that. So upon fertilization, the intracellular calcium ion concentration of the egg increase in a very greater amount. The calcium ion responsible for the cortical granule reaction are stored in the endoplasmic reticulum of the egg. After the calcium ion level rise in the sea urchin egg, its intracellular pH also increases. That means there is a overall basic condition inside a egg. Last step is the IP3 is formed at the site of a sperm entry in a sea urchin. So we are going to understand this whole line with the help of a diagram. This is a diagram of the IP3 DAG pathway, and I have already discussed this pathway in the cell signaling lecture. So if you have not seen that video, I will mention the link in the description box so that you can watch this, because all the basics behind this process is totally same that we have discussed in the cell signaling lecture. So what happened actually, there is a production of IP3 and it is going to act as a in a primary mechanism for the release of the calcium from intracellular storage. So the concept is that inside the egg there is a presence of a endoplasmic reticulum, and endoplasmic reticulum is regarded as the storage of calcium. So what happened actually, there is a binding of a sperm with a egg, okay, and the sperm receptor is present on the egg membrane. Now it is believed that the egg membrane, like the receptor that is present on the egg membrane, it is having a nature of the receptor tyrosine kinase, which is going to activate the PLC, okay, phospholipase C enzyme that is present in the plasma membrane of a egg. Now this PLC is going to break the PIP2 into the DAG and the IP3. Okay, DAG and IP3 both is going to act as a secondary messenger. Now this IP3 is having the you can say that it is having the capability to bind to the receptor that is present on the endoplasmic reticulum, and after the binding of this IP3 to the receptor that is present on the surface of the endoplasmic reticulum, there will be the release of a high amount of a calcium inside a egg cell. So this in this way there is an increase in the intracellular calcium, and this calcium is going to help in the exocytosis of the cortical granules. So I have mentioned the two figure. First is just showing you the overall representation of the IP3 DAG pathway, and the second figure is going to show you that how this IP3 DAG pathway is working with in case of a sperm and egg. So I have mentioned that after the production of the intracellular calcium, it is going to help for the exocytosis of the cortical granules. And after that, the whole process, what is going to take place, we have already studied in the previous slide. One thing you have to note that the presence of IP3 is very important, and it is regarded that the production of the IP3 is at the same location where the sperm makes an entry inside a cell. And from that location, the IP3 is going to enter inside the ER of a cell, and then there will be the production of a calcium that is going to make a exocytosis of the cortical granules. So the concept is that if you will inhibit the IP3, there will be the no production of a calcium. Hence, the slow blockage of polyspermy will not going to take place, as there is no one to activate the exocytosis of a cortical granules. Now let's talk about the second factor that is responsible behind the activation of a cortical granules. So from the figure of IP3 DAG pathway, you can see that the DAG is able to activate the sodium and hydrogen exchange pump. And by the activation of the sodium and hydrogen exchange pump, there is a efflux of hydrogen outside the egg, and there is an influx of sodium inside the egg. Due to this influx of a sodium, there is a increase in the pH of a egg. That means there is a increase in the alkalinity or the basicity of the egg environment, and this is going to make a positive response for activating the cortical granules exocytosis. So the activation of this sodium hydrogen exchange pump is done by both the DAG as well as the calcium ion. This is the overall representation that how the fast block of polyspermy and slow block of polyspermy is going to occur, what is the role of a IP3, and what will going to happen after the release of a calcium. So you can go through this pathway, you can pause the video and you can go through it. Although I have already explained each and every term that has been used here, but I have arranged all the different points in the same video so that you can connect the each and every process with each other. This is some experimental condition that is important from exam point of view. So the first experimental condition was that the polyspermy can be induced if sea urchin egg are artificially supplied with the electrical current that keeps their membrane potential negative. Okay, so in the first slide we have seen that for the fertilization to occur, there should be a negative membrane potential of our egg. So if we provide the artificial condition for which is going to favor the negative membrane potential of a egg, obviously at that time there will be the occurrence of a polyspermy. All these experimental cases has been proved by different type of experiment. You can go through the Gilbert and there you will going to study a lot of things that is has been accepted by different scientists, and they have performed many different experiment to prove the different condition. The second experimental case is that the fertilization can be prevented entirely by artificially keeping the membrane potential of a egg positive. So suppose there is a no fusion between the egg and the sperm, but if somehow with some experimental condition, if you are successful in keeping the overall membrane potential of a egg in a positive way, then obviously in the absence of a sperm also there will be the absence of a fertilization, because the egg will be regarded as the positively charged, so no sperm is going to act on that. The third experimental case is that the fast block of polyspermy can also be stopped by lowering the concentration of a sodium in the water. So as we have seen that the initial source of entry of sodium in our egg is from the seawater. So in artificial condition, if we lower the concentration of a sodium from the water, automatically there will be the prevention of a polyspermy. The last experimental case is that the inhibition of a IP3 synthesis prevent the calcium release, while injected IP3 can release the sequestered calcium ion in a sea urchin egg, leading to the cortical granule reaction. So this line I have already explained in the previous slide, that if there is absence of a IP3, there will be the no release of a calcium, and hence there will be the no activation of a cortical granules. And in the second condition, if you provide this IP3 from the artificial manner or from the outside, and you will inject that IP3 inside the egg, at that time there will be the again a naturally release of a calcium ion, and that is going to help in the cortical granule reaction. Now let's come to the question. The first question is from 2013 June. Polyspermy result when two or more sperm fertilized an egg. It is usually lethal since it results in blastomere with the different number and the type of chromosome. Many species therefore have two blocks to a polyspermy, the fast block and the slow block. So in the case of sea urchin, so they have given you a total of 4 statements. You have to pick any 2 statements that is correct in the case of a sea urchin. So in case of sea urchin, there is total of 2 type of prevention of a polyspermy which used to occur. First is the fast block and second is the slow block. So you have to pick a correct statement that is going to designate the fast block definition and the slow block definition. So the first option is that the fast block is the immediate and cause the egg membrane resting potential to rise, which does not allow the sperm to fuse with the egg and is mediated by an influx of a sodium ion. So this option is totally correct because it is just representing the correct step of fast block of polyspermy. Second statement is that the fast block is immediate and cause the egg membrane resting potential to rise, which does not allow the sperm to fuse with egg and is mediated by an efflux of a sodium ion. So this statement is not correct, because in this step they have mentioned about the efflux of sodium. That means there will be the removal of a sodium from inside environment of a egg. So there is not at all this kind of a event which used to take place. So this B statement is totally wrong. The C statement is the slow block or the cortical granule reaction is mediated by a calcium ion. So this option is totally correct because the cortical granule reaction mainly used to take place with the help of a calcium ion, and this is also known as a slow block. The fourth option is the slow block or the cortical granule reaction is mediated by a potassium ion, and this option is totally wrong because there is a no involvement of the potassium ion. So the correct combination of an answer is the A and C, which is the first answer. The next question is from CSIR Net 2016. Consider the following event which occur during the fertilization of sea urchin egg. And in the last line, they have told that which of the above statement or statements is not true. So you have to pick the wrong statement for the fertilization event of a sea urchin. So let's eliminate it. So the first statement is that the resect or the spirect are the peptide released from the egg jelly and help in the sperm attraction. So this statement is totally correct, because sometimes we call it as a resect or the spirect, and this used to act as a chemoattractant that has been secreted by the egg, and this used to attract the sperm. So this statement is totally correct. The B statement is binding on acrosomal protein interact in a species specific manner with egg. So there is nothing to explain in this line, because this line is totally correct. Third statement is a respiratory burst occur during the cross linking of fertilization envelope, where a calcium dependent increase in oxygen level is observed. So first of all, this statement is totally wrong, because there is a no cross linking of the fertilization envelope. Okay, fertilization envelope used to occur in the very last step of the slow block of polyspermy. And second thing is that there is a no calcium dependent increase in the oxygen level. Okay, in the IP3 diagram, we have seen that there is an influx of the sodium ion due to the help of a calcium. Okay, so we can say that there is an increase in the intracellular sodium ion, not the oxygen level. So we are considering this C statement is totally wrong. Now let's move to the D statement. The IP3, which is formed at the site of a sperm entry, sequester calcium, leading to the cortical granule exocytosis. So first of all, the meaning of sequester is to hide. Okay, and the function of the IP3 is not to hide the calcium. It used to bring the calcium from ER to the cytoplasm. So IP3 used to play a role to expose the calcium inside a cell, not to sequester the calcium. So the first line is correct, that the IP3 which is formed at the site of a sperm entry, but the second line is totally wrong, that it used to sequester the calcium, leading to the cortical granule exocytosis, because the cortical granule exocytosis required the calcium. Okay, and if there is a sequestering of a calcium, how the calcium is going to be available for the cortical granule reaction? So this statement is also wrong. So according to our concept and knowledge, I will consider that the C and D, both this statement is totally wrong. Okay, and in the official answer key of CSIR net, they have marked a asterisk instead of a answer. So they are saying that if you attend any option for this question, you are going to get a marks. So although we should know that how to eliminate a wrong answer, because it is totally depend on our concept, it doesn't mean that this question is wrong. We are going to take it in a very lenient way. Obviously we should know that how to eliminate a wrong answer. So from my side, there should be like C and D option, both is wrong, and there is no option which is going to represent the C and D combination. Now let's move to the next question, which is from CSI net 2017, and this question is very interesting. When I have attended this paper, I think I have took only 1 minute to solve this question. Okay, it seems to be complex, but if you read, you will understand that this question is very easy. So the question is, fertilization in sea urchin involve the interaction of a sperm binding with its receptor, that is EBR1, a 350 kDa glycoprotein on the egg vitelline membrane. The plot given below show the status of the membrane potential and level of a EBR1, sodium ion and potassium ion in an unfertilized egg. So the first graph that is given below in front of you is for the unfertilized egg. Okay, and the blank bar is going to show you the level of a EBR1, which is acting as a receptor and is present on the egg vitelline membrane. The black bar is going to represent the concentration or the level of a sodium ion in unfertilized egg. The shaded bar is going to represent the concentration and level of a potassium ion in an unfertilized egg. Okay, and now everything is clear that what is the level of EBR1, sodium ion and potassium ion inside a unfertilized egg. So there is a very high level of a EBR1 receptor inside a unfertilized egg, and like this is very normal, because the receptor is always there on the surface of a vitelline membrane. Next is the presence of a sodium ion, which is obviously will be very low in an unfertilized egg. And next is the calcium ion, which is supposed to be present in a like little more higher amount, and this is only factor that is responsible behind the negative charge of a egg membrane. Now let's see the condition of a question. Which one of the following graph best represent the condition within an egg 1 to 3 seconds after the fertilization? So if you like right now, I think most of you have understand the meaning of this question, that after the 1 to 3 seconds of fertilization, obviously there will be the occurrence of a fast block of polyspermy, because this is a condition of the fast block of polyspermy. And if we recall the different step of fast block of polyspermy, it is going to include the entry of a sodium from the outside environment to the inside of a egg cell. Next is the there will be the little bit exit of a potassium ion from the egg. And the if we talk about the third factor, that is the EBR1, there will be the no effect in the level of a EBR1 because it is acting as a receptor. And like if we have seen in the step of a fast block that there is a no involvement or there is a no harm on the EBR1 receptor. So we already know this three condition. Now let's match this three condition with a given option. So we have seen all the different conditions of the fast block of polyspermy, that there will be the increase in the sodium concentration, there will be the decrease in the potassium concentration or the little bit decrease in the potassium concentration, and there will be the no effect in the EBR1 after the fertilization. So if you will see the all graph, the first, second and the fourth graph is showing the very high level of a potassium ion concentration. Okay, and this has no sense why there will be the increase in the potassium ion when there is a occurrence of a fast block of polyspermy. Okay, so in this way I have easily eliminated the first, second and the fourth option. Okay, and I will now left with only a one option, that is the third option, but we have to tally this third option with whatever the condition which we have concluded till now. So if we talk about the third condition, so EBR1 is going to show a high expression because there is a no effect on EBR1 after the fertilization. The second is that there is a high level of a sodium ion concentration inside a cell due to the fast block of polyspermy. And the third graph, third bar which you are going to see is the bar for the potassium ion concentration. So obviously there will be the little bit decrease in the potassium ion concentration. Okay, so in this way we have concluded that the best answer is the option 3. There is always a different kind of a hint in this type of a question. Like here there was a hint like why there will be the sudden increase in the potassium ion concentration. So if someone know that there is a no or the little effect in a potassium ion concentration, and that also in a negative way, not in a positive way, so that student is going to eliminate this first, second and the fourth option very easily. With this, we have completed the topic of prevention of polyspermy in sea urchin. I know this video has become a little bit long, but we can't do anything. These all topics are very time consuming. So if you like this video, then don't forget to hit that like button. You can share this video with your friends who are preparing for the CSI and NET examination so that it would be beneficial for them also. And if you are new to my channel, you can subscribe my channel for getting more different videos on CSI and NET, GATE and many different examination. And along with that, don't forget to hit the bell icon just beside the subscribe button so that you will be notified with my each and every new video uploads.